So in our last section of notes, we looked at the history of DNA and the experiments that were used to eventually find the structure of the DNA molecule and to learn that DNA was the genetic material that was being passed on through the generations. Today what we're going to focus on is do actually a quick review of the DNA structure and really focus on the function of DNA and specifically how DNA is replicated. So looking at the DNA structure, it says the double helix structure revolves around the orientation of the 4 different nucleotides. So a couple of things we need to look at here. When you are examining the DNA model, a couple of things you should notice. First of all is that we have a very specific pattern as to which nitrogen bases pair with each other. When you're looking at these nitrogen bases, you should be able to see that A and T always pair together and G and C always pair together. Both of these are creating what's known as a complementary base pair. Something else that you should notice on this molecule is that each of the A's and G's are what's known as double-ringed or purines, and then your C's and your T's are pyrimidines. So this is a purine and this is a pyrimidine. Between them you should also see a different bonding pattern. When thymine and adenine bond together, they bond using 2 hydrogen bonds. When guanine and cytosine bond together, they use 3 hydrogen bonds. Because a purine and a pyrimidine always pair together, and again here a purine always pairing with a pyrimidine pair together, this actually creates what's known as a uniform diameter or a width, and we'll go ahead and say due to purine to pyrimidine pairing. The other thing, and so just to sum up what we've just said, is then we have a double ring. That's going to be our purines. And our single rings are going to be our pyrimidines. Another thing you might notice here are the words anti-parallel going along the sides of the DNA molecule. And anti-parallel is talking about how it runs in opposite directions. And we're actually talking about the direction of the sugar, of the deoxyribose. The way that this molecule, the deoxyribose, is numbered is actually based on which carbon is facing in that direction. And so we have a 3' to 5' direction. And so if you notice here, you have carbon 1, 2, 3, 4, and then 5 is right here. So we are 5'. Runs in opposite direction due to the carbon. And then on the same thing, you'll see the 3rd carbon is actually in this direction. Okay, so 5' to 3' and 3' to 5'. And that'll be more important when we talk about replication. And then the last thing that we mentioned here is that the hydrogen bonds are actually weak individually, but strong as a whole. So when you combine all of these hydrogen bonds together, they do make a very strong molecule, DNA molecule, and the 2 sides of the DNA ladder are unlikely to break apart. However, with the help of an enzyme, it can individually break those hydrogen bonds. But once you've reviewed the structure of DNA, what we need to look at now is the overview. What is the purpose of talking about DNA replication in the molecule? You should be able to remember that DNA holds the code to build any number of proteins, And this protein code comes from a gene. And that might be needed by any cell at any given time. We're really going to focus here on DNA replication, where the DNA is copied. Why do we need to copy DNA? Or why do we need to replicate it? We will need to replicate DNA during mitosis, and this is one reason, where we are just producing 2 new cells from the individual cell. We might also need to copy DNA during meiosis, where we're actually copying the DNA to later on make some genetically different daughter cells. Additionally, what else is the DNA used for? We can also use the DNA to make what's known as mRNA during the process of transcription. And just to remind ourselves now, or to mention it now, during transcription, all we're doing is rewriting the DNA. Then later on during translation, this is where we actually assemble the amino acid chain to eventually build our protein, reminding us that our protein actually forms our traits. They can also become enzymes to speed up reactions. And some examples of some traits would be pigments, such as eye color that we've talked about. Oops, sorry about that. Okay. So going from there, we would just want to very quickly review one final experiment. And this experiment actually explains to us how did they figure out the process of DNA replication. Okay, last time we focused on, well, how did we figure out that DNA is important, that it's the genetic material. And so today we want to look at how was the DNA identified as being what's known as semi-conservative. So we had 2 scientists, Messelson and Stahl, and they used, again, some radioactive isotopes to identify how DNA molecules replicate. When they were using isotopes, just as a reminder to you guys as what is an isotope,